Good morning, students. In my previous video, we have discussed the roadmap for this chapter, that is your molecular basis of inheritance. Now, in this video, I am going to discuss the first chapter or first topic of this chapter, that is your search for genetic material. Now, before going to the search for the genetic material, we must know about the characters of that particular material. What should be the characters of this genetic material so that we can search it? If it is present inside the body, inside the cell, or inside any part, then we must know this character. Now, what should be the character of this genetic material? The characters of the genetic material must be, the first one, replication. The molecule must replicate by its own. It can produce, suppose it is a molecule. Now this molecule can produce the same molecule by its own. Okay. So otherwise, how can the molecule send the message to the next generation? If the molecule is present there in the parent, the same molecule must be sent to their offspring to express their character. So that molecule must have the capacity to replicate itself by its own. So that is the most important character. Now the next important character, that is the stability. The molecule must be stable generation after generation, so that the character must be stable. Otherwise, there will be variation, change in the character in the next generation, and in the next generation, the variations will be much more. So it may create a problem. So that is why the molecule must be stable generation after generation. But third important thing, if it is 100% stable, then also it may cause problem, because if there is a need of any change, if there will be any adaptation is required for the environmental changes, then at that time the organism may face problem. So that is why there must be the chance of some mutability, or that is the chance of mutation, so that it can change in this molecule, so that some characters may be developed in the organism to survive in the nature. So that is also important. Now next one, it must have this property that is called expressivity. Suppose this is one molecule, no doubt, but this molecule must express the character. So that is why the characters are expressed by this molecule. So that is why the molecule has the character to express this molecule in the form of character. So considering these four important characters, so many scientists, they try to find a particular molecule present inside the body, inside the cell, but the most successful experiment conducted by Griffith, that is called transformation experiment. Transformation experiment. And this transformation experiment was conducted by Frederick Griffith in 1900. F. Griffith in 1928. In his experiment, he took the mice as the specimen and another bacteria, that is called streptococcus or diplococcus bacteria, streptococcus bacteria, and this streptococcus bacteria, which is commonly called streptococcus pneumonia, and this streptococcus pneumonia, it causes pneumonia disease. Pneumonia disease in mice. But this streptococcus pneumonia, they are of two variants. ये दो तरह के होते हैं. Two variants are there. The first one, that is your smooth variant or smooth variety, and another one, that is the rough variety. The smooth variety is having a slimy smooth coat over this bacteria. ये smooth coat एक रहता है. So that is why it is a smooth coating bacteria. And another one, there is no smooth coating, that is why it is a raw bacteria. So this smooth bacteria, it is a virulent means if this smooth bacteria is inoculated or injected into the mice, then it causes the pneumonia fever. But this raw variety, it is a non-virulent type. Non-virulent means if it is inoculated or injected into the mice, then it does not cause any disease. So what are the main findings of his experiment? From this experiment, he actually found the following thing. First one, when he injected the mice with the smooth variety, then it suffered from disease, and when he injected the raw variety, it does not cause any disease. Now, on his first experiment, he took the smooth variety, okay, and when this smooth variety of bacteria, when it is inoculated into the mice, then the mice normally die, because the smooth bacteria, it is a virulent type. It is a virulent type and it suffers from pneumonia and the mice die. Now second one, when he injected the raw bacteria to the mice, then the mice does not die but it survived, because this raw bacteria, it is a non-virulent type. Non-virulent means it does not have the potentiality to kill the mice. It does not have the smooth coating over it. But when in his third experiment, he took a smooth bacteria, or this smooth bacteria, but by killing it, giving heat. These are called heat-killed bacteria. When he injected the heat-killed bacteria to the mice, then the mice survive or it live. It does not die. ये नहीं मरता है. Now, next, when he injected the smooth bacteria, which are heat killed, along with the raw bacteria, which are non-virulent type, so the raw variety into the mice, then surprisingly he found that the mice die. Now, when he inoculated the smooth bacteria independently and the raw bacteria independently, then both survive. But when the heat-killed smooth variety and raw variety both are combined together, inoculated into the mice, then the mice die. Now, he could not prove that biochemically. How this happened? Actually, he dissected the dead mice. Surprisingly, he found some live S or live smooth bacteria. So he found the live smooth bacteria there. Actually, the bacteria were heat killed. No live bacteria was there. But when this heat killed bacteria and raw variety bacteria, heat killed smooth variety and raw variety both combining together, introduced or injected into the mice, how this smooth bacteria is formed there? From where the smooth bacteria comes from? So this smooth bacteria, actually it is transformed from a raw bacteria. It is the raw bacteria transformed into the smooth form. Now, a question arises, now what is actually, which molecule or which factor which is there in the smooth variety or a dead smooth variety which is responsible for the transformation of the rough variety into the smooth variety. Now, this explanation was given by three famous scientists, Avery, MacLeod and McCarthy. Avery, MacLeod and McCarthy gave the biochemical explanation to the Griffith experiment. And in this Griffith experiment, actually the heat-killed bacteria or heat-killed smooth strain bacteria, it actually gives certain substances or it normally transfers certain substances to the raw bacteria so that this raw bacteria converted into the smooth form of bacteria. Now, what are the different molecules which are there inside the smooth variety? And according to their analysis, they found that there were some DNA molecules inside this, some RNA molecules, some protein molecules and lipid molecules. There are so many other molecules also present there in the cell of heat-killed smooth strain bacteria. Now, they try to analyze which molecule out of these molecules is actually responsible for the transformation process. Now, first of all, they give certain enzymes to digest these molecules. They gave the DNase molecule. This DNase molecule or DNase enzyme, you can say, this DNase enzyme, it digests the DNA and it denatures the DNA and the DNA becomes functionless. So that is why when they added the DNase molecule into the medium, they found that the mice were alive. That means there is no smooth variety of bacteria or smooth strain of bacteria. Now next, they added the RNase enzyme into the medium. When they added the RNase enzyme, RNase digests the RNA molecule, but it was found that the live S form or smooth variety was found and the mice died. Again, they added the protease enzyme to it, and this protease enzyme, they digest the protein molecules present in the medium, and due to which what happened, the smooth variety of bacteria also observed there and the mice died. Again, they added the lipase. It is another enzyme, and this lipase enzyme digests the lipid molecule, and they found that the same smooth variety is there and the mice were dead. Again, there were so many other molecules which were there and gave the enzymes to them, and these enzymes, they digest those molecules and found that the smooth variety of bacteria was still there and the mice died. From this, it was concluded that the DNA molecule which is actually responsible for this transformation process, because this DNA molecule, when it was digested, then there is no smooth variety and the mice survived. So that is why from this explanation it was concluded that DNA is the genetic material for the transformation of the characters from one generation to another. So this concluded that the DNA is the genetic material. Now, the second famous experiment conducted by Hershey and Chase, and they conducted the experiment on the bacteriophage. The bacteriophage are the viruses which normally infect the bacteria, and there are two types of bacteriophage. He cultured in two different culture medium, and the first one, which was normally cultured in the S35 culture medium. That means the culture medium containing the sulfur 35. The sulfur 35 is a radioactive chemical, and which substance is normally incorporated into the protein coat, because the sulfur is the one of the important constituent of the protein. Now, another culture medium which contains the phosphorus 32, and this phosphorus 32, it is a radioactive substance which is incorporated into the DNA molecule, because the DNA molecule contains the phosphorus. So that is why the culture medium which contains the phosphorus. Now the bacteriophage, they have the DNA with this P32 radioactive substance. Now, when these bacteria, these bacteriophages, they now infected into the E. coli bacteria, or when you can say the E. coli bacteria is added to this culture medium, then automatically these S35 radioactive bacteriophages, they attack the E. coli bacteria, and when they attack the E. coli bacteria, the DNA content which are there inside the bacteriophage, they enter into the cell of the E. coli. Now, in the second culture medium, here the DNA molecule, which is now the radioactive having P32, is entered here into the cell. Now second, after that, now blending is done. Blending means the agitation or sacking of the culture medium is done, and due to sacking, the outer coating, the outer portion which was attached to the bacterial cell, they were separated from the bacteria. The detachment of the bacteriophages occur from the E. coli bacterial cell wall. Then third phase, when the centrifugation is done, what happens? The supernatant contains the outer substances of the cell, and this supernatant of this first culture medium, it normally no radioactive sulfur in the cell. So radioactive substance was not there in the supernatant, but here the radioactive phosphorus normally in the cell. So what happens here? No radioactive substance in the, or that is the S35 you can say, in the cell. This is the cell medium, and this is the cell or E. coli cell, and after centrifugation, the radioactive substance or sulfur 35 was normally not there in the cell. And second, in the supernatant, what is there? The radioactive sulfur is only present in the supernatant. Supernatant means the fluid which is outer to the cell. Then the sulfur is only found in the supernatant, means it is there in the coat of the bacteriophage. Here what happens? The radioactive phosphorus is present in the cell. That means it is incorporated into the genetic system of the bacteria. Now the supernatant, which does not contain or no radioactive phosphorus, is there in the outer coating. So it normally confirms that the DNA molecule is entered into the cell of the bacteria, and here this DNA molecule actually responsible for the expression of the characters in the next generation bacteriophage. So here only in the second case, here the P32 entered into the cell, and this P32 DNA core, which is responsible for the expression of the next generation bacteriophages. So that is why from this experiment, Hershey and Chase concluded that this DNA which entered into the cell actually responsible for the expression of the character in the next generation. So from this they concluded that DNA is the genetic material. So from these two famous experiments, finally it was concluded that DNA is the genetic material. Now I hope it will be helpful. Then please do like it and subscribe it for next videos.